Hi, everybody, and welcome to Neuroscientists Talk Shop, UTSA's neuroscience research podcast. Today is September 18th, 2025, and we're talking with Ricardo Mozzachiodi. See, I'm better at it when I'm practicing ahead of time. And Mozzachiodi. There, I got it right. And Ricardo is professor in the Department of Life Sciences at Texas A&M Corpus Christi, and his lab studies the neuronal circuits, synaptic connections, molecular mechanisms that subserve learning of some relatively simple behaviors in a relatively simple organism, Aplysia, the sea hare. And there's a wonderful literature on learning in this particular creature, and a lot of what we know about the cellular mechanism of learning was first discovered there, and a lot of new things continue to be discovered in that preparation. And I hope we can review some of those accomplishments as well as discuss some of Ricardo's recent findings. So hi, Ricardo.
Hi, nice to meet you. And thank you for having me on the podcast.
Also joining us is Alfonso Apicella, our go-to cellular neurophysiologist at UTSA and longtime podcaster. Hi, Alfonso.
Hi, Charlie. Hi, Ricardo.
And I'm your host, Charlie Wilson. So Ricardo, could you start by just giving us a short summary of the logic that led historically to the study of learning in Aplysia and the strengths that people hope to gain, and then sort of what we got out of it?
Sure. So the story of Aplysia, it's a mollusk. And the story behind this animal is that some scientists in the late 50s, they, particularly Eric Kandel, he is a neuropsychiatrist, he wanted to study the human brain and the pathologies associated with memory loss. But obviously, it was very difficult. You cannot really do in humans. And so he started answering this question on rats. And so, he was trying to record from brain cells in the rat brain with very low success at that time, and there was not really any signature that could tell you, oh, this is a memory cell versus not a memory cell. So apparently, at a seminar, a lady presented work conducted in France using a snail for the advantage of having very large cells and very easy to track connections. So that animal was Aplysia. Now, there's Aplysia in every ocean. So there's an Aplysia in the Atlantic Ocean close to France. There's several in the Mediterranean, and there's one in the California Ocean. And so the idea that Eric Kandel had is that why not using this simple animal that must learn something, finding a way to track simple reflexes. And if you get these two things, you can understand the basis of memory at the cellular level. So they had to go through, demonstrated that simple forms of memory happen in this animal, and then demonstrated that they could actually track the cells at the cellular level. And my doctoral advisor actually had the fortune to be in New York at the time that this effort was made, and he contributed to tracking the mechanisms that are happening in this animal. So the idea was to have something really simple, but an animal that still has the need to learn, the need to form memories, and potentially even to form long-term memory, the memory that lasts for a very long time. And what is very important is that even if this guy is very far away from us, so far, the cellular mechanism, the molecular mechanism, are very, very conserved to the one that we have.
And of course, the behavioral forms of plasticity that were being studied are ones that we share also.
Exactly.
Sensitization, habituation, and simple associative learning.
Exactly. And they specifically studied with things very simple.
Ricardo, I have a question. When you say simple, because, probably is not a common knowledge anymore, but there was a C. elegans person not long time ago where this animal has only 300 O2 neurons. Fire Mello did a massive work, with electron microscopy to map all the connectivity of this simple nervous system. But after so many years, when we're talking about simple nervous system, those days were almost scared because it's not that simple. what do we mean with the simple, correct?
Yes, and we mean simpler.
Okay.
And it's a very good point. And it's a point that sometimes I correct myself when I tell the story, because the main goal was to have an organism that has reduced the variable as much as possible. We have what in the human brain about 80 billion cells. A rat brain, millions. But if you have a system in which you can have a limited number of cells, but the animal perform functions that are vital for survival, and also, as Charles mentioned, has to learn basic things, you can combine them. And it's a point of start. You can have something to the place. And then obviously you continue with this experiment and you realize that the system is not simple at all.
One of the benefits, I think, just as an outsider reading the literature of Aplysia compared maybe to C. elegans, was that they were looking at a reflex. And so they weren't trying to ask, how does Aplysia do everything that it does? Which is, C. elegans folks were kind of thinking, how does C. elegans do everything, right, all at once? And in this case, they were just saying, well, if I puff some water on the gill, the siphon will withdraw, right? And so you don't have to figure out how every neuron in the nervous system contributes to that. I needed to find the sensory neuron that was carrying that information. And there's like one. Is that right?
Yeah.
It seemed like there was just one. So that was really helpful. If there had been 100 of those, it would have made it more complicated.
There is only one neuron in Aplysia that does this.
That carries the particular sensory information from what they were doing in that one.
There were very limited numbers, yes.
Yeah.
Okay,
Yeah.
And then there's a motor neuron that drives the withdrawal thing. And there's a synapse, a group of synapses made by that one sensory neuron onto that motor neuron. And so that, there could have been 1 trillion neurons in Aplysia, but if you could get that amount of information, you could dissect it. I think at least as important as the structure of Aplysia was the clarity of the question being asked.
And also, I think ultimately, with the research moving on, was the ability to not even looking at these cells in the circuit, but ultimately to take the cells out, put them in a petri dish, and reconstruct the circuit. So bottom line, you can ultimately see the behavior just with 2 cells in a petri dish. And now, obviously, we're talking about several decades ago, and there's many, many models. Everybody has their favorite model. Drosophila is a fantastic model for genetics. Accessing the neurons, it's not easy. So everybody has their favorite.
Yeah.
And advantages and disadvantages.
Yeah. Well, practically, the advantage of being able to take the nervous system out of the animal and mimic the behavioral environment by stimulating a nerve and recording from one meant at that time, compared to recording in vivo in a mammal, where any tiny little thing causes you to lose your recording, here you have a stable preparation that's doing everything that you were interested in, that the whole animal is doing, and isn't doing any of the things you didn't want to do. So, I know I want to ask you about some of your recent work, but I wonder if you would say something about the fundamental pieces of the learning, of the cellular mechanism of the learning story, and bring us up to the point where you're at in your current work.
Well, one is that a great advantage of using a system like this mollusk is that you can actually work at pretty much 3 different levels, potentially 4. So you can study the behavior of the animal, that you can study something at the circuit level, bottom line, the neurons, and how they work as a team, as the output of the team being a reflex or a more complex behavior like biting. And then you can even go deeper and look at the individual cells. Ultimately, harvest the cell from the brain and put them in a petri dish alone. So that gives you those many different levels, and they're not separate. They're interconnecting. So you can go from the highest to the lowest and then to the middle one, etc. And this allows you to do manipulations that potentially may not be able to do in the animal. For example, you can track the activity of a cell before and after a treatment in the isolated nervous system. You can inject a molecule in the cell body of the cell and see how the changes happen, something that obviously you cannot do in the entire animal. But as long as you know that you're not just poking a cell, you know exactly what that cell does, you know what the cells play a role in the circuit and in the behavior, that actually you can have a lot of connection for the dots.
And the cells have wonderful names.
Yes,
with letters and numbers in them.
Yes, and it's true. Yes, and when we show students the map, we make the joke that the cells come with numbers. So if you open the brain, you see the numbers. So of course does.
And you always get where B51 is, correct?
Yes. And now there's
one of the cell name was B51.
Exactly, and it was just simply because they were B stands for buccal. This is the region of the brain that controls feeding. And they started with the two largest cells and they called them B1 and B2. They're giant. The joke is that the bigger they are, the little they do. And they're giant neuroendocrine cells. And then you go for B3, B4, and ultimately. But it's not just not naming it. Ultimately, science were able to associate the physiology of that cell to a specific function.
So every cell has a name, or were people just naming the cells as they were learning what they did?
Yeah, I think they started numbering them anatomically.
Just completely anatomically.
Yes, and they're still unknown cells. My former postdoctoral advisor, who also does computational neuroscience, his name is Jack Byrne at the University of Texas McGovern Medical School, they were doing a computation of the feeding network. And at one point, their computation was great, but it could not be perfect unless you add one cell that did not exist. So they called it X. A few years later, they found the cell.
It's just like the discovery of Uranus.
Exactly. It's another planet.
Yeah.
Exactly.
That's great. That's science at work. So I think the key thing that came of it was that most people know about is that the actual mechanism of memory turned out to be just changes in the excitability of neurons and the strength of synaptic connections. And even just one synaptic connection could be enough to actually do something. And that those changes were being done by neuromodulators that act through G protein coupled pathways.
Exactly.
And that discovery propagated out and is now sort of universally used in discussions of cellular mechanism of memory in every species.
And I think what is important is that there's a conserved mechanism. So bottom line, when they found that there was a pathway in the nervous system of a simple animals, it can be C. elegans, it can be Drosophila, it can be Aplysia, it's not just them. The mammalian brain may have it too. And so bottom line, these essential basic mechanisms are universally conserved. And they can apply to us. Obviously, we are more complex. And, there's a lot of price with complexity. But those essential basic little building blocks are still there for us.
So can I test another principle that I extract when I read this? I don't know if it's really true. It's the pathways that we normally think about, which have transmitters like GABA and glutamate and do fast synaptic transmission, and you can say, there's the synapse, there's the presynaptic side, there's the postsynaptic side, there's the synaptic cleft. Those are not the ones that control learning. They're the ones that carry the message, but the molecules that control the changes and the strength of those things are not released in that very punctate way. They are modulators that are more or less acting like local hormones or something. I don't know how local, maybe really, really local, but they're definitely not just acting at synapses. And this seemed to be, is this still true? I mean, in all of the things that you've learned in Aplysia, is this holding up, that these things are not being carried by the fast synaptic transmitters?
Well,
and does it make sense that it would be like that?
It does, or it doesn't. It depends. So, for example, yes, in speaking of Aplysia, the first thing that was observed is that the changes that happen may not due to the communication between cell A and cell B. Now, you can still have plasticity of cell A and cell B, for example, in habituation, which is a very simple form of non-associative learning. Basically, you habituate, your brain learns to disregard something neutral. It could be just A and B alone. But for other forms, you need a third party. And that third party, as you mentioned, could be a little far away neuron releasing serotonin, as an example. But that molecule can be acting A, locally, at the connection, but also globally. For example, in some animals, that molecule is released in the bloodstream. And so it can virtually go from one place to another. And that was an important thing because there's some evidence that some forms of memory, they do require for the long-term persistence in the animal that molecule is actually traveling as a modulator.
So the temporal precision of learning, I don't know, psychologists have names for this. I don't necessarily know what it is, but how close together that one stimulus has to be to the response or to the other stimulus in order for them to become associated. That stuff is determined largely by the properties of the modulator.
It does.
Is that true?
It is true. In some cases, it depends. Now, there can be what we call activity-dependent forms of plasticity, which is just a circuit that interacts. But in many, many cases, it's the modulator. There are some systems in other creatures, like crustaceans, in which you have very small cluster of cells in which there are more modulators than neurons. So there's like 50, 60 neurons and tons of modulators in the form of peptides. And people are still trying to grasp what is the purpose.
That's a puzzle.
It's a puzzle, exactly. And but it's fascinating because, again, you cannot explain everything just with the connectivity. You need these guys.
Yeah, but today also, when we were looking at the serotonin and your two types of different behavior, correct?
Yes.
One was the aversive and the other was the appetitive behavior.
Yes.
You also, at some point, you mentioned that there were two different pathways, correct? One that was PKA and the other that was PKG dependent.
Yes.
I was curious if you can tell us more how these PKA and PKG act on the glutamate release, correct? Because we also, in the mammalian field, we think that they almost, in a very simplistic way, they have an antagonist function, correct? The PKA increase the glutamate release, the PKG lower the glutamate release. Is the same mechanism applied to the Aplysia and in the control of the aversive versus appetitive behavior?
Let's start with saying what PKA.
Protein kinase.
Say something about what those are.
Yes.
So Alfonso points out to the fact that there's a lot of kinases, and kinases means there are basically cells of enzyme and molecules that change something else. Now, in the case of the data that I showed today, we discovered that for these very elementary forms of learning called sensitization, in the long term, there was an historical cascade. And the historical means being there for a long time. And it's a serotonin to PKA.
We mean it being known for a long time.
Being known.
Because it was there for even longer.
Yeah, of course. Before us. Yeah. What my work showed is that there seems to be an analog. And now the question was, how do they interact, as you said? Are they synergistic? Are there like parallel lanes? Or one influence the other? So we have one little piece of data that I was not able to show in my lecture today that seems to point that serotonin precedes NO. So bottom line, NO is a downstream of serotonin.
So NO gets released into the extracellular space and acts as a modulator that changes synaptic transmission. But the NO gets released by the presence of serotonin?
That's what we seem to see. We don't know. So there's a big debate in the plasticity of what a molecule has what we call anterograde or retrograde effect. So bottom line, you can have neuron A influencing B, but there's always a possibility that something happens in neuron B affecting it.
This is why it's so hard to find out what's cause and what's the effect.
Mm-hmm. Exactly. So my data do not point to a specific location, but we have some just behavioral data that shows that the two pathways are not parallel. One is upstream of the other. So, and it's just a work in progress. So every day, every time you add one piece of the puzzle, but then you got more.
So earlier you mentioned something about spatial scale, which, I was thinking about temporal scale before, but spatial scale is important too, because whatever's happening in the vicinity of where the modulator gets released, that's what's eligible to be changed. And if you're not close enough, then you won't get changed.
Exactly.
And so different modulators may have different sort of diffusabilities or distributions of release or something that causes their spatial scale to be different. Is there a way for us to see that? Can we know that? Like if we're recording from a neuron here and a neuron there, and we see this one changes and that one doesn't?
It's a really good question. There's a lot of imaging tools that we have these days. I don't have in my lab. I would like to go deeper. So bottom line sometimes is really going one level deeper to see what can happen. NO is a gas, nitric oxide, which means that basically the guy can pass through membranes. He doesn't need to a receptor on the membrane. He just has to molecules in the cytoplasm of the cell. Mm-hmm. So there's a lot of possibility for it. A big question for something like this is exactly, does it act locally because as a gas quickly disappear, or can stay longer? It's a very fascinating question.
Scavenged by molecules in the blood or something.
Exactly. Yeah. So for serotonin, if I may, again, computation and real experiment in Aplysia, they show people went through a careful analysis of many pulses of serotonin you need to release to get changes. Because the idea is that if you have a continuous action, probably that doesn't give you a very long memory. So one pulse of serotonin gives you something short. Two pulses of serotonin still relatively short, which it means too short to the animal. 3, 4, and there seems to be really a one point, the summation of events. Also put them close in time in which you open something new. And a message can be, for example, instead of just staying in the in the cytoplasm, can travel to the nucleus. And then you get a long-term change.
So in mammals, the neurons that, like Alfonso studies and me, the ones that release modulators like acetylcholine or dopamine, their axons have this enormous, huge, furry ball. Whereas the ones that are carrying the discrete messages tend to have very spatially localized things. I always wonder, you see, here's a neuron whose axonal field goes 2 millimeters, and here's another one that goes only a few microns, and we can see that, but we can't really interpret that or see what that means.
Can you do imaging? Can you image some?
People have been doing acetylcholine.
Correct. They have biosensor right now. Correct. But they're called biosensor. You can see that. I had another question that was intriguing me from really an animal point of view. Correct. You say that particular aversive behavior, the memory, correct, it only lasts 72 hours. For an animal, correct me if I'm wrong, I was learning, it only lives 1 year, correct? Only have a memory only for 3 days is not decremental. I'm thinking about the animal in the wild, correct? Let's say aversive behavior is a predator touching me, and then 72 hours I have no idea. Basically, I'm a prey, correct, that a predator can attack me all the time. Should be another mechanism that should keep this memory longer in order to the animal to defend itself?
There are, so everything always starts as some kind of artificial approach, because what you do, you play with stimulation. You play with the number and you play with timing. So, for example, one thing that I didn't have a chance to mention today is that in several systems, people explore different paradigms. So, for example, one shock usually gives you only a short-term memory, maybe a couple of hours. Come back the next day, the animal, and this is not only Aplysia, can do Drosophila, the animal has forgotten. You do 4 in a day, you get memory that lasts about 48 hours and it's gone at 72. But you can go further. You can, for example, do what we call a long-term protocol. It's 4 days, 4 shocks, 4 trials a day. And that gives you the animal a memory that lasts weeks. So what you go in is it is possible for the animal to remember longer.
But then, since we were saying that the molecular machinery, the underlying molecular machinery that is leading to this can be similar between the Aplysia and the mammalian brain, how we explain that sometimes, for us, a single event leads to this memory they're going to last us forever? Why is not happening in the Aplysia, or what are the underlying molecular pathways that are different or similar?
It's a very good question, and my tentative answer is that it may depend on the stimulus. So, for example, they have what they call light bulb memory, right? So, bottom line, it's one exposure, something so massive that leaves you a memory that lasts forever. Your wedding day. You don't need to get married 4 times spaced to remember your wedding day. So, there's something that probably is really powerful, but it depends on the stimulus. So I can't say that Aplysia may do it. It's just that you need to find something that it's so powerful that activate these cascades, a cascades that is not just simply a random surge and then everybody drop back, but you can compress the timing and do something.
Also, you're looking at a behavior that you wouldn't really want to inhibit forever. Like if you're looking at feeding, and something has caused me to feed less, if that thing goes away, I need to start eating again.
And if that's what we see. So, for example, the protocol that I just mentioned. So if you apply 4 days of training, 4 tries a day, you get a memory for defenses that last for weeks. Feeding, only 24 hours. Actually, sorry, I apologize. 48 hours. So bottom line, at that point, for something so motivational driven like feeding, shocking the animal 4 times or shocking the animal 16 times for feeding makes no difference. The animal needs to resume eating. And so bottom line, that tells you that even in the dynamic, there's much more complexity. And some of them we may not even know. So...
yeah, I wonder about, like, a lot of the examples that we use when we think about single trial learning, taste aversion or something like that, are sort of episodic. Memories are like, I went to the beach with my family, it was such a great day. And I wonder if, like, Aplysia have that kind of episodic memory. I mean, I don't want to make it sound like they're not a good example of everything, but they may not be a perfect example of every kind of human experience, but just the things that they are good at. That, I don't know.
They may or may not. Maybe I don't know. Maybe evolutionarily speaking. So it's the same thing of emotion, right? So we have a specific center. They don't. But they still need to feel fear. They need to experience fear because for them, fear is survival. It's not only fear, but the memory for fear, because otherwise you become a meal. So even if they don't have a centralized center like the amygdala that is critical for emotional fear learning, they may still function. And it's just that they have much less cells.
Is the same circuit that you are studying with the shock on the body, is the same one when they are in the surface and you touch them and they go deep, they touch them to the bottom and they start to crawl?
I'm not sure.
Yeah, I was watching this video that was so fascinating. They were very young. I never, for me the picture is always Eric Candela grabbing a big Aplysia, correct? But yes. I saw it, they can be really tiny. They can fit in your hand. I saw today. And there was this video where they were on the surface, swimming, and then these scientists touch them, and then basically, once again, I thought that almost like, electrical stimulating on the side, they dive, they go down to the bottom, and they start to crawl. I say, wow, that is really fascinating. I was wondering if this is the same underlying circuit or network of cells. They do the same behavior.
It's possible. Now, these guys don't do too much, but they definitely do more than what I showed today. I mean, they crawl. They don't swim. This species doesn't swim. Other do. So they crawl. They gallop.
They gallop?
Yeah, they have like a movement that looks like galloping. So they like projecting up, then reattach and projecting up, or they just simply crawl on the surface. They wave their heads looking for food. So they have different behaviors. You can basically train everything. You can do classic conditioning, differential conditioning. So bottom line, you can condition an Aplysia by touching a tentacle and shocking the body, and you do that on the left tentacle, and so it's the left tentacle that's going to be conditioned versus the right one. And that was a big paper that Candela and Carew published many decades ago, showing that a simple animal like this can do what we call differential conditioning. So they're capable of doing sophisticated things, still with a relatively small amount of cells.
Well, it's a great example of how you pick the species that allows you to do the experiment you want to do, and the payoff can be huge.
I hope so. I believe so. Yeah, and there's many other systems. There was a Danish physiologist. His name was August Krogh. Probably some of you know about the Krogh principle. He was studying lymphatic system. He received the Nobel Prize at the early 1900s. But he wrote this sentence that I put in my lectures that says that for every animal in nature, there must be one that is good for your purpose. And it can be anything, but it can be the one helping you answering some questions.
Yeah, it's a sort of difference in philosophy of doing the experiment, whether you think I have to do it on this animal because this is the animal that everybody does
Correct.
this stuff on, or whether you think, how could I benefit from trying something different?
One of the things that I think, and maybe, correct, maybe I'm wrong, or maybe not, but Eric Kandel picked these animals because he wants to study the molecular pathway, correct, of why Aplysia at that moment was for him the perfect animal model to look at the molecule, correct?
Well, first of all, the circuitry.
Correct. But then you won't go down to the molecule,
yes,
correct?
Yes.
So
Why?
the question is,
yes,
how far ahead was he thinking?
Correct.
So you're saying he was probably thinking all the way down to
the crab, correct?
Yes.
How? Correct. What do you think? Historical. Can you give us a summary, or can you get in the Eric Kandel head and see how?
Well, it depends if he's listening or not. Well, I really like the story because it shows you, first of all, somebody who has a really very big creativity, and just not looking at one aspect, but as you said, thinking forward. There was nothing at that time. And the goal was to understand disease in humans. But before that, you need to try to understand, even before you try to understand a disease, you need to understand how things work normally, right? Otherwise, you don't know how to distinguish between the pathology and the normal physiology. And pick up something that could give you an idea of simplicity. His first attempt was mice and rats, but it was like one recording every 3 weeks of one neuron. It was very...
I read his classic papers on hippocampus with Spencer.
Yes.
Mm-hmm. They were very well received and good. He was the only one that was disappointed with them, I think.
I think in a sense, yes. But maybe because exactly it felt that he couldn't move forward to the level of analysis. And I don't know, maybe that seminar light up a bulb of using a model that still needs to function, still need to remember, and in theory, evolutionary might be still linked to us. And but then you're right. I mean, initially they had to go to demonstrate that the animal learns. How do you know that it happens, right? So demonstrate learns form, simple form, and then characterize the circuits that control the behavior that show learning. And it's also an enormous effort.
Yeah.
The important point is that he was able to collect a powerful team of people that within a few years, it's Operation Warp Speed, for learning enough for the messenger RNA. So, and then what I find pretty cool is that, yes, there's a tracking of all the cascades, but then can they function in a mammal? And he demonstrated that it works in the hippocampus.
Well, thank you.
He went back to the hippocampus.
Yeah, he did. He went back to the hippocampus. So thank you, Ricardo. That's a great story.
Thank you very much.
Inspiring for us, and we should maybe think more creatively about our experiments as well. And thanks, Alfonso.
Thank you, sir.
And this has been Neuroscientists Talk Shop.